I want to thank everybody for joining us for this poster presentation on Efavirenz-mediated drug-induced liver injury. And it's being presented by Diane Longo. And so, Diane, you go ahead and take it away when you're ready.
All right, great. Yeah, thank you so much. Today I'm going to be presenting a poster. It was previously presented at the ACAP conference in 2020, and going to describe using DILIsym, a quantitative systems toxicology model, to represent Efavirenz-mediated drug-induced liver injury. And a little bit small here, so I'll try and zoom in as we go. So just as a bit of background, efavirenz is a compound that's used to treat HIV infections. And this compound has been associated with a low frequency of liver enzyme elevations in treated patients. So we wanted to use DILIsym to see if we could represent the hepatotoxicity mechanisms that may be underlying the clinically observed liver signals. So to do this in DILIsym, one of the first steps that we take is to start to collect in vitro assay data for 3 primary hepatotoxicity mechanisms, specifically bile acid transport inhibition, mitochondrial dysfunction, and oxidative stress. We've seen in DILIsym across over 75 compounds that with these 3 mechanisms, roughly 80% of the time, we're able to successfully predict the DILI liabilities for various compounds. So we use the in vitro toxicity data to determine toxicity parameters for the compound of interest. And in addition, we use physiologically based pharmacokinetic models to predict the liver exposure for the compound. And then we combined the predicted exposure with the appropriate hepatotoxicity mechanisms in simulated populations, or what we call SimPops, that include variability both in hepatotoxicity mechanisms as well as in exposure. And this allows us to simulate the in vivo response in humans, and thus to predict the frequency of liver injury. So I'll shift over to look more closely at some of the in vitro assay results. On the right side of the screen, the first plot on the left shows in vitro data that was used to assess the capability of efavirenz to induce mitochondrial dysfunction. So this data was collected in HepG2 cells at 1 hour using the Seahorse XF analyzer assay and used to measure the oxygen consumption rate, or OCR. And we can see that with efavirenz, there's a dose-dependent decrease in the OCR. This can be suggesting that efavirenz is acting as an electron transport chain inhibitor. So we use this data to try to determine the ETC inhibition parameter values for efavirenz. And we do this by starting in mitosym, which is a standalone model of mitochondrial bioenergetics. And we first optimize the ETC inhibition parameters in mitosym. That's shown with the red dots in the simulation here, where you can see that the simulated reduction in OCR by design is recapitulating the reduction in OCR seen in the data. And so once we have the parameter for ETC inhibition in mitosym, we can translate that parameter value into DILIsym. The plot on the right shows data that was used to assess the ability of efavirenz to elicit oxidative stress. This data was collected in HepG2 cells at 24 hours and assessed using a high content imaging assay. And the black dots showing the data show that there is a dose-dependent increase in oxidative stress with efavirenz. So we use this data to determine the efavirenz-mediated oxidative stress generation parameters in DILIsym. And again, by design, the red dots you can see for the simulated increase are recapitulating the observed dose-dependent increase in oxidative stress that we see in the data. So in addition to the mitochondrial data and the oxidative stress data, we also collected data to assess the ability of efavirenz to inhibit 4 bile acid transporters: BSEP, NTCP, MRP3, and MRP4. And we're not showing the raw data here, but in these transporter assays, we saw that efavirenz could inhibit 3 of these transporters: BSEP, NTCP, and MRP4. And so the IC50 values from the in vitro assay results were used as inputs into DILIsym for inhibition of these transporters by efavirenz. So next, we combined the toxicity parameters determined from the in vitro data with predicted exposure. And so for this study, we used a GastroPlus PBPK model that was actually just described in a previous session that had been developed by my colleague Inger Darling. And so we used that PBPK representation to predict liver exposure for efavirenz for 600 mg QD dosing for 12 weeks. So with the predicted liver exposure and the appropriate toxicity parameter values, we then simulated the toxicity response for efavirenz in SymPops. And for this study, we used SymPops with 285 normal healthy volunteer individuals. And in the simulations, we saw that following administration of 600 mg QD efavirenz for 12 weeks, there were 1 to 2% of the simulated individuals with ALT elevations greater than 5 times the upper limit of normal. So this low frequency of predicted ALT elevations is consistent with what's observed clinically, where patients treated with efavirenz have had 1% to 8% with ALT elevations greater than 5 times the upper limit of normal. So in conclusion, with DILIsym, we were able to correctly predict the infrequent hepatotoxicity for efavirenz. This was done by combining the in vitro mechanistic data for 3 hepatotoxicity mechanisms, predicted liver compound exposure coming from a PBPK representation, and SymPops with biological variability in both underlying hepatotoxicity mechanisms and in the exposure. So this study provides further support that DILIsym represents a powerful tool for assessing the potential DILI liability of new compounds. And in addition, now that we have this representation of efavirenz in DILIsym, we could in future studies use this to predict DILI drug-drug interactions between efavirenz and any other compounds that efavirenz may be co-administered with. And I'll pause here. I'm not sure if there's any time for questions or if we move on to
Diane, you've got time for a couple of questions. One thing I just want to say, I love situations like this where you're combining 2 different models together to really strengthen your results. That's not a requirement with DILIsym, is it? It's simply more of a bonus for this particular project that you had a PBPK model that you could integrate in with your DILIsym.
Yeah, so exactly. With DILIsym, we can use any source for the predicted exposure. More and more, we are using GastroPlus PBPK representations as the direct input. We use specified data to specify these concentrations, and more often now we are using GastroPlus PBPK representations to predict the exposure, although alternate sources of exposure could be used as input into the model.
Nice. We do have a question coming in of, was any non-clinical study used to confirm the ability of the PBPK model to predict liver exposure? I think that question may be a little more applicable to Ingers, but I think I can expand it for you in that, do you know if any non-clinical or clinical data even was leveraged to kind of validate the DILIsym outputs?
Well, not for efavirenz, but we have many exemplar compounds in DILIsym. Some of the one of the primary ones being acetaminophen, where there's just a wealth of data. So as I mentioned, we've at this point now run over 75 compounds through DILIsym, many of them which did have various multiple sources of clinical data that we could use to continue to validate and refine the model.
Nice.
And
nice,
I should mention that with efavirenz, although it wasn't, we already knew that there was some liver toxicity with efavirenz. It wasn't, we didn't optimize to that toxicity response. This was in a way a pure prediction where we're putting in the inputs on exposure and toxicity. And so predicting the liver toxicity that ultimately did align with the clinical data, but it certainly wasn't optimized to that predicted liver toxicity.
Yeah, I like that, we had Inger's recorded presentation about the PBPK model leading into your SimPop and DILIsym model. And you did a wonderful job even segueing me into the next speaker, which is Brett, talking about the quantitative systems toxicology for acetaminophen.
Great, thanks.
So with that, I think we can go ahead and adjourn a little early to give people time to go to the next talk with Brett. That should start in a few moments. I encourage everyone to please leave the current session and then join the next one with Brett, and I will see you there. Thank you very much, Diane.
Thank you.